Hello, everyone. Welcome to our very last lab recording for this semester. And in this one, this is the second part of the nervous system. We're going to be focusing on the brain and the cranial nerves. So, as we get into this, as we begin our exploration of the brain, at about 6 weeks of development, you have five what is called cephalonic divisions. And these five cephalonic divisions will be out at birth, not roughly about 9 months into your development. You will be born with a brain that has all the regions that just need to develop into a fully functional adult brain. So these five cephalonic divisions develop into some structures. The first region that we want to see now, when I teach my lecture, I begin with forebrain, midbrain, hindbrain, and those develop into these five cephalonic divisions, and then the five cephalonic divisions develop into the things next to it. So the telencephalon will develop into the cerebrum. Think, you need a cerebrum to tell me about it. Diencephalon, to put the di in diencephalon, we want the thalamus and hypothalamus, which make most of it. So the thalamus and hypothalamus, and then pituitary gland make that diencephalon. Thalamus and hypothalamus put the di in diencephalon because they are making the bulk of it. Pituitary is kind of small. Mesencephalon, midbrain. Midbrain, it's a mass in the midbrain. There's a lot of things where visual and auditory centers come in and they are sent to appropriate regions. For example, when you see like light in this area here, you're going to help adjust your pupil's diameter when light enters the eye. So there's a visual processing center there. And then also auditory processing centers. For example, if I make a really loud noise, there's regions in this that are going to tense the muscles up to cause you to tense up due to a loud noise. But it's important that we test those visual things. That's why usually a pupillary reflex is a very common thing to study the integrity of one's midbrain because it's also crucial to maintain consciousness. Now, the metencephalon, where the cerebellum and the pons is located, or cerebellum and pons becomes from this area, metencephalon, think about the cerebellum is actually attached to the back of the pons, so they have met. Metencephalon, cerebellum and pons is what that becomes. The myencephalon will ultimately develop into the medulla oblongata. Think me, oh my medulla oblongata. The medulla oblongata, which means middle and oblong shape, when you combine the medulla oblongata, pons, and midbrain, this creates the brainstem. Now, remember, the text materials from my notes are going to be how I make multiple choice questions, the 20 multiple choice questions on your lab midterm or your lab final. Sorry. Now, as we go on, you can see, as I said, at 3 weeks, you actually have three swellings. These three become five. The five become the regions of the brain at birth that will then further develop. Now, as I want to help you guys understand, we talk about the cerebrum. The telencephalon makes the largest part of the cerebrum. Then we talk about the diencephalon, which is mostly thalamus and hypothalamus, but also pituitary. So this puts the di in diencephalon as the thalamus and the hypothalamus. The midbrain, the pons, and the medulla oblongata. Remember, midbrain, pons, and medulla oblongata come together and make the brain stem. And the cerebellum is attached to the back of the pons, hence they have metencephalon. Okay, now as we go on to the cerebrum, remember by far cerebrum is going to be the largest. It comes from the telencephalon. One needs a cerebrum because if you can't tell me about it if you don't have conscious thought and intellectual functions. So the telencephalon becomes the cerebrum. Now, on this, it is extremely well folded. There will be gyri and sulci that help to increase surface area of cerebral cortex. The gyri are elevations and the shallow depressions are the sulci. Now, gyri stick up, and a good way to remember this, if somebody jumped on top of a table and just started gyrating, that would probably stick out. The elevated ridges stick out. They help to increase surface area of cerebral cortex. We're going to talk about cerebral cortex as that thin layer of gray matter on the outside of the brain. The sulci, this is shallow depressions. And then there's also very deep grooves. You can think of them as cracks in the brain called fissures. And then we're going to see the gray and white matter are very different in the brain. Now remember, the gray matter of the spinal cord is internal, but gray matter here is found in the outer cerebral cortex and then deep in the brain in what's called basal nuclei. The basal nuclei of the brain is going to be kind of associated to this area of things like your lenticular nucleus and that kind of stuff. It's deeper nuclei. They're integral to functionalities of the brain as well. Now, white matter is either going to be under the cerebral cortex or surrounding the basal nuclei. Now, the surrounding basal nuclei, like the internal and external capsule associated to the lenticular nucleus. The hemispheres of the brain, there are two. There's a left hemisphere of the cerebrum and a right hemisphere of the cerebrum. And separating the left and right hemispheres of the cerebrum is a longitudinal fissure. Now, I've got to use diagrams only. So far, this is this is Tuesday. I might not actually be able to get on campus until Thursday. It's been today. It's been a very hectic, busy day. It's actually why I'm recording so late today. Is I just got back from the polls to vote. So. All right, so let's go take a look at some diagrams. So we could see that gyri are the outer folds that go up, and sulci are the smaller depressions, and fissures, like this right here, is a very deep fissure. And also, what we could see is there's a left hemisphere and a right hemisphere. Now, diagrammatically, you could see the two hemispheres. There is a right and a left hemisphere separated by the longitudinal fissure, separates the two hemispheres together. Now, one of the things I always test on is the lobes of the cerebral cortex. Now, to help you remember these, we all know Sigmund Freud was a guy who studied the human brain, studied psychology, father of psychoanalysis. And Freud was kind of probably, he always thought people had, it's like we all know what a Freudian slip is. That's when you say one thing, but you mean your mother. He had hang-ups about like moms and things like that. So Freud tore pants off. Freud, frontal lobe, tore temporal lobe, pants, parietal lobe, off, occipital lobe. Those are the four lobes for cerebral cortex. Freud tore pants off. I don't know why it's so hard to say. Now, frontal lobe, one of the things I will always ask you guys on this exam is what these different lobes do. And one of the things I want to make sure you guys understand is a frontal lobe is, first off, the primary place where you voluntarily control your skeletal muscles. If you're going to voluntarily control skeletal muscles, there's a portion of the frontal lobe, more specifically, we're going to learn that is called the precentral gyrus, and it is mapped out to control specific parts of the body. But so voluntary control of skeletal muscles, but also cognition, that's thinking, reasoning, that's thinking through your actions, and logic is understanding things like applying and understanding and using reason. It's like logic is basically the application of reason. Reasoning is using cognition. Cognition is thinking. So what happens is the frontal lobe, reasoning and logic are what's going to allow you to think through the long-term impacts of what you do. And actually, when you're young, it's still not fully developed. Your prefrontal cortex isn't fully developed to about 17, 18 years of age. Now, given that, you're driving at 16. Well, your prefrontal cortex hasn't fully formed, which is a major component to allow you to actually determine the long-term impacts of your actions. Like if you were to drive recklessly, you might kill somebody or yourself or hurt somebody. And, okay. Now, parietal lobe. This is the primary sensory cortex. Now, what does it do? This is where most of the body senses come from. I like to say the primary somatosensory cortex because this consciously perceives touch, pressure, vibration, pain, temperature, and taste. Touch, if I tap you on the skin, you'll feel that. Pressure, if I squeeze and push into that. Vibration, your phone goes off in your pocket. Pain, something like a bee sting or a sticky with a needle. Or very, very hot temperature burns you. Pain. temperature and taste. Now notice, some tastes can be painful, some temperatures can be painful. For example, I found very sour tastes painful. So actually, now some people find, of course, very cold things painful, very hot things painful. Now I love spicy foods, but cannot stand thermally hot things when I eat. But I love spicy foods. We were eating some we ate some we had some tacos we made, and we always have our different hot sauces out when we make our tacos, and I'm putting ghost pepper sauce on my tacos, and nobody else wanted it. I was like, man, I said, man, it kicks hard, but it's gone like fast. I mean, like now occipital lobe, it's at the back of the skull, and this functions in conscious perception of visual stimuli. Temporal lobe is your auditory cortex and olfactory cortex. What does that mean? It means it consciously perceives the senses of hearing, auditory, and olfaction, smell. So, a lot of times, when what's interesting, if you put magnets on one's temporal lobes, they will hear and smell things that aren't there. And a lot of times, too, you may notice certain sounds and certain smells affect you in very strong ways, especially certain smells. It's like if a lot of people will drive through in the country and they'll smell cow poo out in the cow pastures by farms and going, shoo, that stinks. I kind of find it personally pleasant because I grew up as a country boy. I grew up in a place called Possum Trot Ridge, if that tells you anything. And so grew up rather poor. We raised pigs on our property and things like that. So just the smell of manure, kind of, to me, it's more of a pleasant odor to me. Now, as we go through it, our frontal lobe is all this kind of reddish, this dark red and this light red here. The dark red is actually the area where you will send motor commands out of. That's that mapped area. It's called the precentral gyrus. And so all of this, dark red and lighter red, is frontal lobe. And then here we have this kind of more yellowish is temporal lobe. Parietal lobe is this dark blue and light blue area, and the green is the occipital lobe. Occipital lobe deals only with vision. Now, let me explain this to you guys. When I say it primarily does visual stimuli, it's what allows you the conscious perception of visual stimuli. It isn't the only part of the brain that receives visual information. Visual information is always also relayed to the midbrain and also relayed to other parts of the body to adjust muscle tone and things like that. It goes to and affects the precentral gyrus here. So there are a lot of impacts that visual has. Primary somatic sensory here mostly goes here. Now, of course, on this, auditory and smell, olfaction, and auditory hearing goes here to this lobe, but that's not the only place that information gets. But this is where you can consciously perceive these things. It must go to these areas on the brain to consciously be aware of it. So, or leave this area to consciously have control of it. So, the cerebrum. We talked about the major anatomy. Now let's get into some more nitpicks. Let's start, the first thing I want to talk about is the central sulcus. It is the dividing line from the frontal lobe to the parietal lobe. Now, in front of the central sulcus, there is a gyrus called the precentral gyrus. It is part of the frontal lobe and serves as our primary motor cortex, which is a very fascinating area. There's a little mapped out area on this called a motor homunculus. Homunculus means little man. And the postcentral gyrus is behind the central sulcus, posterior to it, and it's actually on the parietal lobe. And it is the site of the primary sensory cortex. This is the primary somatosensory cortex, where, like, if you poke your finger, there's a very specific part of the postcentral gyrus that information has to get to so that you will feel that as fingertip. If you stub your toe, there's a very particular part of that it must get to. And I'll show you what I mean by these. Now, what I'm going to do is show you guys a motor and sensory homunculus. Homunculus. Now, you can actually, if you want to, guys, by the way, those of you guys who have Dr. Lockhart, tell him I said that he's Homunculus Rex, king of the little people. Okay. Now, so here, if it's motor, if you want to control the toes, you got to get the information from about here. If you want to control your knee, from about there. If you want to control your shoulders, it's got to leave the areas underneath this. If I poke your thumb, it's going to get to here to tell you that it came from the thumb. If I'm going to hit you in your head, it's got to get to this little fold here for you to know that was the head. It actually has to get to the area there to actually be known about it. This is actually where it must get to actually be consciously perceived as that part of the body. Sometimes they go to the wrong place. Now, that also means, like, if you don't have a leg, this is how phantom limb pain happens. The part of the brain, the part of the postcentral gyrus that senses the legs are still there. And sometimes sensory information gets to it, so you feel something that isn't there because of the part of the brain, the part of the postcentral gyrus that actually is determined to feel that is still present, though the limb is gone. So you get phantom limb pain. Pretty cool, huh? Now, lateral sulcus. This is going to separate your frontal and temporal lobes. If you pull the lateral sulcus apart, you will see a deep part of the cerebral cortex under that lateral sulcus called the insula, or insular cortex, some of us will call it, because it's part of the cerebral cortex under that, insular cortex or insula. Now, separating the parietal and occipital lobe is a parieto-occipital sulcus. Now, once we get to the very bottom of our longitudinal fissure, where you connect left and right hemisphere, there's a very big band of white matter there connecting left and right hemispheres. This is a type of commissure. It's called the corpus callosum, the colossal body, the big body that connects the two halves. Now, corpus callosum is needed for left to right communications and needed for high levels of multitasking. And as it turns out, a lot of times females have higher connections than males do. Female brains are generally more connected left and right. Male brains are generally more connected front to back, but this is generalization at best. Okay, not always true for every single person. Now, this left-right connection there is kind of very interesting. Now, the cingulate gyrus is just above that corpus callosum, cingulate gyrus. And then we're going to see olfactory bulb and tract, which is on the bottom of the frontal lobe to carry olfaction to the brain. It's on the inferior side of the frontal lobe, carries our olfactory or smell information. So let's find our central sulcus. Central sulcus is right here separating our frontal lobe from the parietal lobe. And here, anterior to central sulcus is a precentral gyrus. Posterior to central sulcus is a postcentral gyrus. And the primary motor cortex and the primary somatosensory cortex found here. Lateral sulcus found here. If you open it up, you will find an insula or insular cortex there. The parietal occipital sulcus, best seen here, separates parietal from occipital lobe, parieto-occipital sulcus. The corpus callosum is this band of tissue found here. Now, I think for some reason it's not labeled here. It will be labeled somewhere else, but this is the corpus callosum, a band of white matter connecting left and right hemispheres, and superior to it is the cingulate gyrus, located superior to it. But this is corpus callosum. I believe somewhere else it is labeled here, and then also here, and labeled I think that's it. That's the only places it's labeled. And that image, I might go through and do some other edits to my notes just to get that there as well. Now, the cingulate gyrus, we show that. Olfactory bulb and tract is actually right about here. So to find it, we're going to look at this diagram. Here we have our frontal lobe, and inferior to it, we have an olfactory bulb, the terminal end, the end of the olfactory nerve, and the bundle of neurons called the olfactory tract as it goes up to the brain to let me know about smell. Smell affects things like memory. One of the things that will bring back a lot of memories. One thing I remember is when my papa died. After he died and we cleaned out his house, and we were there helping clean out because my grandmother didn't want to live in the house anymore. She lost a husband in it, lost a son in it, and she didn't want to stay there anymore. She wanted to move, be closer to her daughters. And so what we did is we were helping her move, and I can remember we were packing up, and I could just remember as we were moving some stuff out of the bedroom, I got that whiff of Old Spice, Papa's Old Spice, and just tears poured down my face. It just hit emotionally really hard because I can remember him with his arms wrapped around me, teach me how to cast my first Deb Code 33 that he got me, take me fishing, and him teaching me to cast it in our backyard. So just that smell of Old Spice brings that back, and I could just start bawling. I'm about to cry right now, I feel like. So it's these kinds of things here carrying olfactory information, and that really affects the brain. Now, as we go through this, now we're going to go into our diencephalon. Remember, these are diagrams I'm going to use on the test. I can't use models because I couldn't get to them this week. It's been a very rough week. I had a lot to do. Now, diencephalon is actually between, it's on the top of the brainstem. Before you get to the brainstem, it's just above where the brainstem is, and located between your left-right cerebral hemisphere. Mostly you have thalamus and hypothalamus. Now, there is as well an epithalamus, and then we're going to see that there is other things like the pituitary gland and other things associated to the diencephalon. Now, the epithalamus is the roof. The roof of the diencephalon contains the pineal gland, the thalamus, and hypothalamus associated to this. Now, the pineal gland is endocrine. And what does it do? It makes the hormone called melatonin. Remember, hormones go into the bloodstream. Endocrine makes hormones, and endocrine goes endo blood. And this is actually what is involved with your biological clock. Is pineal gland starts to release melatonin as it gets dark. Now, the first night that we had where time had changed and it got dark at 6, I struggled to stay up past 7. I went to lay down about 7. I was just feeling so tired because my biological clock was thrown off. And when you get things like jet lag and stuff like that, it's kind of like, your biological clock's off. Your daily cycles, your yearly cycles, what we call circadian rhythms and circannual rhythms. Now, the thalamus is very large, oval-shaped structure. Looks like 2 eggs kissing in the brain, and it makes 80% of the mass of your diencephalon. Now, the thalamus, I like to describe it, if you guys in America, which all my students are Americans, or at least we live in America, we're all American citizens now, but not everybody listening to this actually will be Americans because I post this on YouTube after I record it for your class. And my one of the things I always do is try to relate this a little bit, and this is something because when I was a kid, after we ate dinner, one of the first things we did is we watched the Andy Griffith Show, then we watched the news, and then we watched Jeopardy, and that was our every single night. That's what we did. Or I believe actually it was Andy Griffith, Jeopardy, and then the news, and then I went and cleaned up for bed. Now, what we would see is the Andy Griffith Show, if you guys are not familiar with it, it's a very old show, but a lot of us probably grew up watching it at some point in our lives, probably saw Andy Griffith, and you guys may remember Barney being in the courthouse, and he'd grab up the phone, and Sarah would answer their lady who was their operator, and he would be like, "Sarah, give me Juanita at the Bluebird Diner," and he would connect her, and Barney would sit because him and Thelma Lou were on the outs again, and he's gonna call up Juanita, and so Sarah is like your thalamus. It's a relay center. So when sensory information comes up to the brain, it says, you need to go to this part of the brain so that I get to the correct area of the primary somatosensory cortex. That's what the thalamus does. And it relays everything except smell. Smell goes to other places. Smell actually has a direct line. Excuse me. It is a very important sense because smell can keep you from eating very rotten foods that can make you very ill. Hypothalamus is the floor of the diencephalon. Now, what we want to associate this is it's just above the optic chiasm where the optic nerves cross. It's located inferior to the thalamus, and I'll talk about it. So here you can see your corpus callosum. Below that, we have here is our hypothalamus, our thalamus, our pineal gland back here. Our pineal gland makes melatonin. Now, as well, you're going to have some other structures. The pituitary gland makes 9 hormones that is endocrine, which are highly regulating hormones. It's the pituitary. Pituitary is just inferior to the hypothalamus here. And the infundibulum is the stalk that holds the pituitary physically to the hypothalamus. Then we're going to have mammillary bodies. The mammillary bodies, there are 2 of them, and they control, like, when it processes like olfactory sensations. So, like, if baby smells mommy's glands on her nipples, the apocrine scent glands on her nipples to initiate chewing, licking, and sucking behaviors. But also, like, when you smell food, you get a little hungry, you start kind of licking and chewing when you smell food. Olfactory stuff stimulate this. And also for babies to feed in sucking and things like that. Optic chiasm is where your optic nerves cross before they go into the brain. AMP2, this is where we'll talk about this. And I always teach the optic chiasm, how the nerves cross, what's going to go on there. So now, as we go through this, you could see a few things here. And all I want to do is the pituitary gland, seen very well here. Here we have our optic chiasm. Then our mammillary bodies, seen here. We have one here and one on the other side. Mamma, they are shaped like breasts. It controls feeding reflexes like licking and swallowing, suckling behavior. Now, this guy here, we're talking about suprachiasmatic nucleus. Very important for when you go to sleep and how you sleep. We'll be talking about paraventricular nucleus and things like that in AMP2. I'll talk about the supraoptic nucleus and the paraventricular nucleus in AMP2 a lot. Now, what I also want to make sure that, so we saw all those, and this is the same image. This is just bigger. So, now the cerebellum is your next largest part. And what it does is it's our little brain, cerebellum. It's our little brain. And the little brain, one of the interesting things it does is, let's say someone tells you how to swing a baseball bat. They say, okay, when you swing a baseball bat, here's how you do it. They show you how to swing it correctly, and you go do it. So in your brain, you intend on swinging it the way that they showed you. And then you do it, and you didn't do it the way you intended to. Now, maybe they tell you, you did it wrong, do this. And so a coach is going to help that cerebellum refine your learned movement patterns. So like when you swing a baseball bat, you have an intention to swing it perfectly, falling through and everything else. But if you don't, your actual movement is wrong, you compare what you intend to do on what you actually do, so the next time it gets better every time. So hopefully you can actually be involved in making a great swing almost every single time. Now, it also is involved in regulating your body's posture and balance. Now, I was born with my cerebellum herniated partly out of the foramen magnum of my skull. If you guys ever get to meet me, you'll see a very big scar in the back of my head. That's because they had to go in through the lamina of some of my cervical vertebrae and chip away part of the occipital bone and remove that and to do a decompression on this. Because if I was ever in an accident, I would have died. But it's so I have very bad balance. You'll see me sometimes just I'll stumble. I can never pass a so-called sobriety check because my balance is crap. And I'm like, but I had a Kahari malformation. They're going like, I don't believe you. And here I am trying on the side of the road, trying to teach neuroscience to a cop. I already, when I was a graduate student, I'll tell you guys a very quick story. When I was a graduate student, I was heading to my where I lived at the time, and I got pulled over by a police officer, and we were collecting tissue samples because we were extracting defense proteins out of the skin of a salamander, and we were using it on ATP synthases in E. coli cells, and hopefully we're going to test it on cancer cells. And so basically, I had this open bottle of 200 proof ethanol in the car, and I got pulled over by a local police officer, and they had me on the side of the road, and I'm trying to show them I have permits for this stuff. I promise you, I'm collecting samples. I'm a scientist. What are you doing out at 3 in the morning with alcohol? Of course, they thought I was up to no good. So, Ed is Pat that if they ever were to test me, I would have failed the sobriety test, would have further probably landed me in jail before somebody said, he's a scientist. Okay. There we go. Sorry, it's a long day and I'm tired and my focus is kind of shot. So, all right. Now let's look at our cerebellum. Our cerebellum has a vermis, and I'm sorry that I left the D bold in there. It shouldn't be bold. There's no reason it's bold. The vermis is worm shaped. It looks like a caterpillar, and then I got meaning worm shaped again. You're like, you don't have to have worm shaped twice, Lefford. I know. It's all right. It's my fault. It's my fault. It divides the left and right hemispheres. It looks like a little caterpillar. Now, there's left and right hemispheres divided by the vermis. There's also a fissure called the primary fissure that separates an anterior and posterior lobe. And then it looks like there's folds on the cerebellar cortex called the folia. Folia, like foliage. I always remember folia. I always remember my French teacher when she would walk in our class, and every day she'd go, un folia de papier vierge, vêtre le nom, les dates, and le jour. We had to get us one sheet of clean paper, put our name, date, all that stuff on there. Le nom, le date, and le jour. Your name, the date, and the day of the week. And the folia is like the leaves, like folds, like sheets of paper. Cerebellar nuclei are going to be deep inside there of gray matter, and then around it is branches of white matter called the arbor vitae, which means tree of life. It looks like a tree, so we call it the tree of life. Every single day almost of my life from 2000, from probably a good part of the mid 2000s was I walked by Arbor Vitae every single day of my life because the science building, I walked between the Arbor Vitae to get in from where I always parked at the, at the university. Now, so we could see our vermis separating the two lobes and then, or the two hemispheres. And then there is a primary fissure separating the anterior lobe in blue and the posterior lobe in this grayish tan oatmeal color. And then you can see the folia, or these little folds that you can see. It looks like if you look at the end of a book. And then in here we can see the cerebellar nucleus surrounded by the thing that looks like a tree called the arbor vitae. Now the brainstem, remember the medulla oblongata, pons, and midbrain is the brainstem. And definitely know that because it always makes a great exam question. Now, the midbrain contains a lot of areas that do unconscious or that also subconscious motor controls or unconscious motor controls, but also things that are maintaining your consciousness, keeping you awake and knowing what's going on. It is between your pons and your diencephalon. Now, the corpora quadrigemina is a very important area. It turns the eyes and the head for a loud noise. Like if a car drives by and backfires next to you, you'll clench your body and your torso in a very loud noise. Now, these things are called colliculi. There are 4 colliculi, so there's actually 2 superior ones and 2 inferior ones. Each individual one is called a colliculus. There are 2 superior colliculi that control the eye and the head for visual information. Superior colliculi, visual information. Inferior colliculi do like the movement of your head and trunk when you hear a very loud noise, like a car backfiring next to you, going down the road, you're walking, car backfires, you hear a gunshot, somebody drops something loud behind you and it startles you. The pons is going to be a connection so that the cerebellum physically attaches to the brainstem here. And information that gets from the medulla oblongata to the cerebrum goes to the pons. And the pons contains these things called the 2 respiratory nuclei. AMP2, we will go into these respiratory nuclei in detail in the lectures. We'll talk about the different nuclei that are there more in detail, the name of them and what they do. The medulla oblongata is the bottom of the brainstem, the inferior-most part, and it is actually attaches to the spinal cord. And it is the choke point for all ascending and descending, all sensory and motor tracts. If it comes through the spinal cord to the brain, and from the brain to the spinal cord, it must go through the medulla oblongata. Now, there are a lot of reflex centers to control the heart, to control breathing, to control your blood vessel diameter, even your vomiting centers. Cardiac control, heart rate, respiratory rhythmicity, breathing rate, vasomotor, whether your blood vessels are dilated or constricted. And I notice I have a typo there. You can slap me later. And also our vomiting centers. Now, the brainstem, remember, the brainstem is midbrain, pons, and medulla oblongata. Okay? Midbrain, pons, and medulla oblongata. Remember, the midbrain has the 2 superior colliculi and inferior colliculi. Superior and inferior colliculi. Remember that there is a medulla oblongata as well. Now, in terms of the colliculi, and then the midbrain, pons, medulla oblongata, and the colliculi. That's all you have to know from these diagrams. Now, the brain ventricles. What is this? Now, this is kind of hard to first describe, but these are actually internal hollow spots in the brain. They are chambers in the brain, and they're filled up with CSF, cerebrospinal fluid. And what happens is these chambers are there because the brain and spinal cord are hollow inside because it developed from a tube. And this tube swelled at the front in 3 places, and then swelled 5 places, and became a brain. And those swellings swelled around a hollow tube, which ends up, as it folds and moves, leaves behind these odd-shaped chambers. There are 2 lateral ventricles, the left and the right, a third ventricle, so basically the first and second is the left and the right. Then the third ventricle and the fourth ventricle are hollow areas formed through here. Now, to separate your 2 lateral ventricles is a sheet of tissue called the septum pellucidum. Septum separates the 2 lateral ventricles. To connect your lateral ventricles to third, there's an interventricular foramen, sometimes called the foramen of Monro. And then there's a cerebral aqueduct, which is also called the mesocephalic aqueduct or aqueduct of midbrain. And this connects our third ventricle to our fourth ventricle, and the fourth ventricle goes out. Now, inside these ventricles, there's a bunch of capillaries and then ependymal cells that not only make cerebrospinal fluid, but help maintain its chemical characteristics, called choroid plexuses. These are very specialized capillaries along with the ependymal cells. You guys talked about those in lecture. I just finished teaching them today in my lecture. Ependymal cells are a type of glial cells, neuroglia, that help produce CSF. Along with the capillary beds, they form your choroid plexuses. Now, you could see your 2 lateral ventricles, 1, 2, and then your third ventricle and your fourth ventricle. Now, lateral ventricles take on a lot of room here. So your two lateral ventricles, your third ventricle, your fourth ventricle. Right here is the interventricular foramen, also called foramen of Monro. And then goes to our third ventricle, down here to our cerebral aqueduct, mesocephalic aqueduct, or aqueduct in midbrain, to our fourth ventricle, which goes on down the spinal cord central canal. And this is all inside your brain. And separating your two lateral ventricles is a septum pellucidum. Septum pellucidum. All right. Now, the last thing we're going to do today is the cranial nerves. Now, one of the things I will do, first off, is test you on name, number, function. Name, number, and function. So let's go over these names, numbers, and functions. And I've got a mnemonic to help you guys learn the names, numbers, and functions of the 12 cranial nerves as they exist in the human brain. So the 12 cranial nerves. Now, these mnemonics that I'm going to do is a visual, then I'll do my regular mnemonics. So what we're going to do is make a head and part of a body with numbers. So there are 12 of them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Now, what we're going to do with these is we are going to take the 12 cranial nerves and make numbers with it. So number 1 is going to be used to make our nose, and that is the olfactory. Olfactory. You can smell the olfactory. Olfactory. Number 2, we're going to make eyes out of that, and that is our optic nerve. And then number 3, if you take a 3 and turn it on its side, it kind of looks like an M, and that is our oculomotor nerve. Number 4 is actually the nerve that controls the superior oblique muscle of the eye, which actually has a trochlea. So it's called the trochlear nerve. Number 5 is our trigeminal, because it has nerve that goes above the eye, below the nose, and at the bottom of the mouth. 3 branches called the, we have the ophthalmic, maxillary, and mandibular divisions. Now, number 6 controls lateral rectus, which abducts the eye. So it's the abducens, or abducent nerve. Number 7. Let's make the sides of our face with 7s. That's the facial nerve. If you ever have to learn the branches of facial nerve, just remember 2 Zanzibar by a motor car. Number 8. Let's make some big old ears on the side of this guy with some 8s, and that's the, I like to call it the vestibulocochlear. Some call it the auditory nerve. Number 9, so what we're going to do with that is I'm going to make this guy stick his tongue out at us. I meant to do this. I'm sorry. I started doing this where I do a 9 like that for the tongue. Sorry. I forgot that 9 for the tongue here to make my tongue. I changed it a little bit. It's called the glossopharyngeal. Number 10 is going to be down here, and that's vagus. Number 11, let's make our shoulders out of 11s. Accessory, or spinal accessory. And number 12, right on the tip of the tongue here, which I used to always put there, by the way, is the hypoglossal. Let me redraw this on the other side here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Now, a good way to remember all these, go ahead and pause and draw it before I move back. Okay. Another good way to remember all these is, obviously, once one takes the anatomy final, very good vodka alleviates headaches. Now, if you're a Harry Potter fan, I also put Harry Potter in the cranial nerves. On they traveled and found Voldemort guarding very ancient Horcruxes. So if you're a big Harry Potter fan, there you go. Now, the other thing is the functions. Functions can be sensory, motor, or both. Now, to remember the functions, I say some say marry money, but my brother says big brains matter more. S equals sensory, M equals motor, and B equals both sensory and motor. Some say marry money, but my brother says big brains matter more. By the way, I do have a YouTube video on this. If you guys go to my YouTube channel, if you're watching this on YouTube, you already know my YouTube channel, but I do have a YouTube video on my YouTube channel that you may already be watching, and will be able to see this video of me drawing out the cranial nerves. I call them the 12 cranial nerves made easy, and somewhere in this, it is here somewhere. Where is my 12 cranial nerves made easy video? Somewhere in this mess, it's here. So, and I know that I have one because it is, there it is, 12 cranial nerves made easy. Definitely check it out. Well worth your time. So one of the things I am going to do is ask you guys the name, number, and function for each of the 12 cranial nerves. And I've given you some really good. Now, on, they traveled and found Voldemort guarding very ancient Horcruxes. And I gave a table where I also gave the Roman numerals. Because some people use Roman numerals. I'm just using Arabic numerals. Now, diagrammatically, the olfactory nerve with the bulb and tract. Number 1. Number 2, optic nerve. Number 3, oculomotor. Number 4, that's IV. IV trochlear, here and here. Number 5, vagus, here and, number 5, trigeminal, sorry, here and here. Sorry about that. It came out, and it's my fault. Number 6, the abducens, found here and here. Number 7, facial, found here and here. Number 8, the auditory or vestibulocochlear nerve, found here and here. And are here and here, yes. Sorry, I had the way it's here. And then our glossopharyngeal here, and then our vagus, which we could see here, and then our accessory here. Okay. So these are non-exam things, but this could be on the exam to show the meninges. These each will be on the exam for sure. Here I've given you a practice of the cranial nerves, and this diagram will be on the test. Draw your spinal cord as a practice, but remember this is your 5-point replacement opportunity. Cranial nerves, here's a practice. Put that on there as well. Guys, this is it. Please don't forget to put your spinal cord drawing on my assignments tab if you guys haven't done it when you go into your class. And log in. What you'll do is we should have, oh, that's lecture. My lecture, not your lecture. But where's I-55? I-55, I-55. There we go. Click on assignments and you will find your spinal cord drawing and can post that there. And it should be ready to rock and roll. It's not. We end, so right now, this week, the third, your final exam starts on the 10th and goes to the 17th, do believe. So right now I'm just going to do that. That is the week before finals. So we'll go ahead and close that, open that up for you guys so you can post it. Okay, guys, that is it. We have finished everything. So sorry that it came out a little bit late. It's just after 6 o'clock now here our time. And it wouldn't have taken as long. I had to try to get to the polls before the polls closed today. And before it got too busy, people got off of work, so I was able to take my lunch break. Instead of going to lunch, I went to the polls so I could vote and get back. My lunch break was not at lunch, so I have such a weird schedule. So anyway, guys, thank you, and I hope I get to see you guys, at least some of you I may get to see a little bit of. But guys, thank you so much for all your hard work. Please feel free to reach out if you need anything from me before the end. Guys, thank you. Have a great day.